Yeah, thank you so much. My name is Vidya Suseela. I'm an associate professor in the Department of Plant and Environmental Sciences at Clemson University. So thanks so much to EMSL for making this work possible, also giving us an opportunity to share our research. So I'll be talking about fine root chemical plasticity in response to nutrient heterogeneity and how that impacts root decomposition. So before delving into the talk, I would really like to acknowledge my co-authors and collaborators from EMSL and also elsewhere, Di Hong Hu, Galia Orr, and Zua Zhu from EMSL, and Luke McCormick, Nishant Tarel, and Peter Kennedy, who are collaborators of this project. Most of this work is done by, let's see, our postdoctoral fellow, Mangja Zia, and she was assisted by the undergraduate student, Courtney Thorpe, who's currently doing PhD at Penn State. Also thank all my funding sources as well. So as sessile organisms, we know that plants are subjected to a lot of environmental stress, both biotic and abiotic. And roots are also subjected to these biotic pests and pathogens, as well as soil physical and chemical changes in the physical and chemical properties or nutrient heterogeneity. So when roots are subjected to these different stress, they adapted to these stress by changing a lot of these root parameters or root traits, like root anatomy, root chemistry, morphology, physiology. A lot of these mycorrhizal associations, a lot of these traits can be changed. So in this study, particularly, we looked at how nutrient heterogeneity or resource heterogeneity affect root traits. So for example, there is changes in phosphorus availability or less availability of phosphorus, how that can affect root traits. We know that the branching intensity can increase, the root exudation pattern can change, it can alter the mycorrhizal association. And so there's a lot of the roots or the plants adapted to these changes by utilizing a lot of plasticity or plastic responses, and mostly physiological and morphological plasticity are well known. But the plants also have plastic responses in small molecules and heteropolymers, which we call it as chemical plasticity. And there is chemical plasticity in the quantity, that is the amount of these chemical compounds and their composition, particularly in heteropolymers. It can alter the number of monomers and the linkages connecting the different monomers. And also where these heteropolymers are located, or can alter the localization as well. So mostly in this study, we looked at the chemical plasticity of fine roots in association with soil resource heterogeneity. So if you're wondering why fine roots, because fine roots are any roots that is less than or equal to 2 millimeters in diameter, and they are very important in plant adaptation as well as from a soil sustainability perspective. So these fine roots are mostly these absorptive roots. They help plant adaptation by altering their morphology, and also they form symbiotic association with mycorrhizal fungi, which then helps in taking up the phosphorus and nitrogen and other nutrients. From a soil sustainability perspective, these fine roots are important because they help in soil carbon sequestration. And so if you're wondering why chemistry is important in terms of fine roots, for both the functions like symbiotic association and for both soil carbon sequestration, it is the chemistry that has an equally or more important role than the morphology of these roots. And currently, we do not know much about the chemical plasticity of fine roots under nutrient heterogeneity. So when I'm talking about chemistry, mostly we are looking into the chemical plasticity of heteropolymers such as tannins and lignins. And so most of these biopolymers, they in live roots or live tissues, they help these tissues to protect against biotic and abiotic stress. And these are the secondary compounds. And so, but once the tissues are dead or senesced, and they still have a biological action because they regulate the microbial enzymes, they regulate decomposition, and hence the biogeochemical cycling. So in this study, mostly we looked into condensed tannins as well as lignins. So with this background, let's look at our objective. Our objective was mainly to understand the effect of different forms of phosphorus availability on the chemical plasticity of fine roots and hence its effect on decomposition. Now, why phosphorus? Because we know that phosphorus is apparently unavailable in soil because mostly it forms iron and aluminum phosphate in acidic soils and calcium phosphate in alkaline soils. So we want to understand how phosphorus availability or different forms of phosphorus affect root morphology. How does it affect root chemical plasticity, mycorrhizal colonization? Both the changes in mycorrhizal colonization and root morphology can in turn affect root chemical plasticity. And now understanding the chemistry is important because it would affect, that's one of the most important parameters that would affect fine root decomposition and hence carbon nutrient cycling. So our experimental approach was we first did a greenhouse study where we grew Quercus rubra and Quercus alba. These are white oak and red oak and white oak for about 1.5 years. And the soil we got the natural soils from the oak forest, mostly the inoculum was from the oak forest. And we had 3 treatments, 3 forms of phosphorus supply. The first one is potassium phosphate, which is a readily available form of phosphorus. Then calcium phosphate, which is less available form of phosphorus, and iron phosphate, even less available form of phosphorus due to its low solubility. So after harvest, we separated them into shoots and roots, and in oak roots or tree roots, we can have like several different orders. And so we separated these different orders, and most of the analysis we did with fine roots, that is any root that is less than or equal to 2 millimeter in diameter. And also we separated into orders. So first 3 orders for morphology analysis, we separated the first 3 orders, and for other analysis, we combined the first 3 orders of fine roots. So we did the morphology using the Winrhizo mycorrhizal association, root anatomy using confocal and flame microscopy. For root chemistry, we concentrated on tannins and lignins, and for localization of lignins, we used the SIMSTOF. For chemical analysis, we used a sequential extraction of tissues to estimate the biologically relevant carbon fraction. So first, the root tissues were extracted with solvents, and we analyzed the tannins, and the sediment remaining was subjected to base hydrolysis and copper oxide oxidation, and that was subjected to the GCMS analysis to understand to get the true lignins, the different monomers of lignins. And then we did a field decomposition study. So what we did was we used these roots and also the soils. We put them in litter bags, and these bags were 100 micron nylon mesh. And we put these bags in the national oak forest at the Clemson experimental forest for 3 years. And today's talk, I'll be mostly concentrating on the first retrieval, that is up to 10 months, and allow it to decompose under a natural condition. So these are the results. So first we will take a look at the biomass and the root shoot ratio. So the total biomass, this is the first panel indicates the red oak, and the second panel is the white oak. The white indicates the potassium phosphate, yellow indicates iron phosphate, and the pink indicates calcium phosphate. And so this is the upper 2 graphs. It is the total biomass, and the lower ones are the shoot root ratio. And you can see that there was no difference between the biomass allocation when supplied with different forms of phosphorus. Okay. So this is the morphology. So the only difference what we saw was that in white oak, those subjected to iron phosphate, in 3rd order roots, there was an increase in the diameter of iron phosphate, in iron phosphate subjected roots. But there was no difference in the 1st and 2nd order roots in terms of root morphology. Again, this was only observed in the white oak, but not in the red oak. But we saw that the resource strategies shifted between the different phosphorus forms. That is, most of these plants subjected to iron phosphate, they had higher mycorrhizal association in both red oak and white oak. And again, this is because iron phosphate is obviously less available form of phosphorus, and plants can provide carbon to the mycorrhizae, and mycorrhizae can easily get the phosphorus from iron phosphate because they're very good at solubilizing the phosphorus, or with the help of mycorrhizal helper bacteria that can solubilize the phosphorus. So the mycorrhizal association really increased with iron phosphate in both red oak and white oak. Now we can take a look at the chemical plasticity. First, we will take a look at lignins. This is the lignin in red oak, milligram per gram. You can see that those roots subjected to iron phosphate, there was a decrease in lignin. And also, when we look at condensed tannins, we also saw that the condensed tannins decreased with iron phosphate and calcium phosphate. So when red oak was subjected to less available forms of phosphorus, the lignins decreased, and also in calcium phosphate, the condensed tannins also decreased. So less available forms really decreased these heteropolymers. That means it increased the carbon quality of these tissues. Whereas in white oak, we saw the trend was kind of opposite, in the sense that in the lignin increased when subjected to calcium phosphate, and in condensed tannins also, the condensed tannins increased with iron phosphate. So in white oak, when less available forms were supplied, this increased the heteropolymers. That means you have lower carbon quality. And so we look at the mycorrhizal association or the root anatomy. You can see that these are the non-mycorrhizal roots, and these non-mycorrhizal roots have a significant endodermis and a large stele. We look at similar mycorrhizal roots, we did not observe that endodermis. So the endodermis was insignificant, a very small stele, and you can see the fungal sheath. And so there was a significant change in the anatomy when there was a mycorrhizal association. And we also noticed that there was a negative correlation between lignin and mycorrhizal colonization. So as mycorrhizal colonization increased, lignins decreased. Mycorrhizal colonization increased, the lignins decreased. This is the SimStop analysis of the same roots. Here what we found was the S lignins were enriched in this endodermis in the non-mycorrhizal roots. So, but that was not observed in the mycorrhizal roots. And our study from previous study from our lab has shown that Mangsha has shown that as the mycorrhizal colonization increased, the secondary compounds really decreased, possibly to increase the mycorrhizal association. So that was the difference in anatomy and chemical plasticity. Now, did it affect the fine root decomposition? So this is the mass remaining in the different mass remaining of these roots in different treatments. And here you can see that as the iron phosphate treated roots in red oak, the quality was higher because of decrease in heteropolymers. There was also increased decomposition. The mass remaining was less in after 10 months compared to potassium phosphate and calcium phosphate. But we really did not observe that trend in white oak, but mostly it was seen in red oak, particularly with iron phosphate. So in summary, what we found in this particular study was that when plants are subjected to nutrient heterogeneity or resource changes in resource form or resource availability, it influences the mycorrhizal association, and that changes the chemical construction of these roots, and that has implications on root decomposition. But it was highly specific to plant identity as well. And so our work basically emphasizes that we need to consider root chemical plasticity and plant identity when investigating the root resource strategy and also biogeochemical or carbon cycle. With this, I would really like to thank EMSL and our collaborators at EMSL. And if you have questions or if you have time for questions. Thank you.